Currently, 3 antibody-drug conjugate targets are FDA approved for the treatment of breast cancer, but many more are being investigated not only for the treatment of breast cancer, but also in other solid cancers. Potentially, some ADCs developed for other cancers could be useful for the treatment of breast cancer. An ideal target should have a high expression in metastatic breast cancer cells with a low intra-patient heterogeneity, but also with a low expression in normal tissue to avoid on-target off-tumor toxicity. We wanted to investigate the expression of these targets in our extensive sample collection of UPTIDER. UPTIDER is our post-mortem tissue donation program for metastatic breast cancer patients. This allows us to extensively sample breast cancer metastases, which would not be possible during life with only limited number of biopsies. We included 30 patients, of which we sent 911 samples for bulk mRNA sequencing, of which 58 primary and treated and 99 normal samples. On these, we looked at the 72 targets that we identified as already investigated in trials. Let's go to our results. We have 3 main findings which I'd like to present today. The first is that we see high expression in metastatic samples of traditional targets such as HER2, TROP2 and HER3, but also of non-traditional targets such as MUC1, LAMP1, EVN1. But when we look at the expression of these targets in normal samples, we also see for some of these high expression in the normal samples as well. When we look at the target with the highest differential expression between the metastatic samples and the normal samples, favoring the metastatic samples, this would be TROP2. But we also see a high difference for HER2, HER3, but also for targets of interest such as Nectin-4 and B7H3. Second, while we see some differences in expression between primary and metastatic samples, we see no differences in expression for the subtypes, so not between the metastasis of triple-negative breast cancer and hormone receptor-positive breast cancer, or between the metastasis of invasive breast cancer of no special type or lobular breast cancer. Third, we also looked at the organ-specific expression in the normal samples and the metastatic samples. Of interest for HER2, we see a higher expression in normal tissue of the heart, but which could explain cardiotoxicity, but not in lung. To conclude, our findings show a lot of new information on currently and already and under targets currently under investigation, and can also help to develop new antibody-drug conjugates, which is very interesting.